All right, so chapter 21 covers the evolution of populations or microevolutions. So the course objective we're covering here is explain how species evolve. So we'll talk about genes and populations. We'll spend a good chunk of time on the Hardy-Weinberg equation or the Hardy-Weinberg equilibrium and its components, and then we'll talk about microevolution and we'll hit natural selection again. So a population, if you remember from Unit 1, is just a group of individuals of the same species that occupy the same environment and can interbreed with one another. A population isn't just people, right? We could have a population of deer mice, or we could have a population of grasshoppers, or we could have a population of violets. Population genetics is the study of genes and genotypes in a particular population, and it helps us understand how genetic variation is going to be related to the phenotypic variation. Remember, the phenotype is the physical expression of the genotype, so it's kind of like what they look like or what genes are expressed. And then the gene pool is all of the alleles for every given gene in a given population. I just put Labradors in the pool there because I thought it was adorable. Really, we probably need some yellow labs in that gene pool too. So the Hardy-Weinberg principle or the Hardy-Weinberg equilibrium says that the allele frequency would remain the same in succeeding generations as long as the following conditions are met. Let's back up though. What's an allele again? Remember, an allele is just a version of a gene. So it's saying the proportion of genes remains the same from one generation to the next as long as there's no mutation, no natural selection, no genetic drift, or an infinitely large population, known gene flow, or a totally isolated population, no mate selection, or random mating. I just like to have the no's all in a row there because it kind of tidies it up a bit. But saying no mate selection is the same as saying random mating occurs. Saying no gene flow happens is the same as saying a population is completely isolated. So what the Hardy-Weinberg principle is saying is that sexual shuffling of alleles in meiosis alone is not enough to alter the allele frequency in a population. Which makes sense, right? If you shuffle a deck of cards, you're not changing the number of cards, you're just kind of changing the order they're in, but you haven't taken out any cards or added any in. So sexual shuffling of alleles alone is not enough to alter the allele frequencies in a population. So let's look at these Hardy-Weinberg conditions. The first one is no mutation, because the mutation could change allele frequency by introducing a new allele. Remember, a mutation is just a change in a nucleotide sequence. So for example, here we've got DNA, mRNA, amino acid sequence. If a mutation occurred and that cytosine was changed to a thymine, that would change the mRNA, which could change the amino acid there. So now we've got a new allele in our population. This is like adding an Uno card into your deck of, standard playing cards. So then with natural selection, the Hardy-Weinberg condition says there can be no natural selection. The super short story on natural selection is that if individuals with certain heritable traits are favored or selected for, the allele frequency can change. For example, if we have this population of crows that prefers to eat green beetles or has an easier time eating green beetles, in future beetle populations, we would expect there to be fewer green beetles and more of those brownish beetles because the brownish beetles did not get eaten as readily, so they survived and reproduced more and passed on their brownish beetle alleles. So the Hardy-Weinberg conditions also say there can be no genetic drift or that populations need to be infinitely large. First of all, can a population be infinitely large? Not really. And then genetic drift is chance events that can alter allele proportions. These events are unrelated to fitness. Fitness in biology is a measure of reproductive success. So this is saying that these events are not related to reproductive success. They're just chance or random events. This could lead to either a loss or fixation of an allele, and this is more likely to occur in small populations, and it will occur faster in small populations because there's less alleles overall to work with. This can also lead to neutral variation, so it could lead to variation that does not affect the phenotype or would be not adaptive. So in this picture below with the same beetles we had with the crows, this person really hates beetles, and so they're stomping on beetles, and they happen to stomp on more green beetles than brown beetles, changing the proportion of beetle color alleles in that population, but it was not due to anything in particular. They were trying to stomp as many beetles as they could, and they happened to stomp on more green beetles. So there's actually two kinds of genetic drift we can talk about. One kind is a bottlenecking event. In bottlenecking events, the population is going to be reduced dramatically and then rebuild. In the picture below, there's a bottle with white beads, yellow beads, and blue beads. That's representing our original population. And then some catastrophic event occurs, and it reduced the population size, and then the surviving population could have a different allele frequency than the original population solely due to random chance there. So lots of times like catastrophic events like out of control fires or flooding, those kinds of things would be an example of a genetic bottleneck. Or we could even have like poaching or extreme amounts of like over harvesting could lead to bottlenecks as well. Founder effect is another type of genetic drift. So in this case, small groups of individuals are going to separate from a larger population and establish a new colony. So this small founding population could have less genetic variation than the original population solely due to random chance. So the allele frequencies in this new population could be different from the original population. So what is genetic drift? Genetic drift is just a change in allele frequencies due to random chance. And the two types of genetic drift we talked about are founder's effect and bottleneck effect. So another of the Hardy-Weinberg conditions is that no gene flow can happen or that populations must be completely isolated. This is because organisms entering or leaving populations bring their alleles with them, so they change the allele frequency. So the two kinds of immigration are a little bit tricky, and people often struggle with this. If they are entering, it's the immigration with an I, and if they're leaving, it's the immigration with an E, so exiting immigration, if that helps at all. Also, this is a very high quality drawing. Gametes can't enter or leave a population because if gametes enter or leave a population, they bring the alleles with them as well. So another of the Hardy-Weinberg conditions is that there can't be mate selection or that mating must be random. So non-random mating can alter allele frequencies, and there's a whole bunch of ways non-random mating occurs. So intrasexual selection is between members of the same sex. So intra within, so within sex selection. So male competition is an example of this. There's also intersexual selection, which is between members of the opposite sex. So like peacocks and their feathers and kind of showy displays are an example of that. There's the picture of a lion. It's actually not a real lion, it's a stuffed lion with interchangeable manes. And there's actually researchers who found out that lionesses, so female lions, prefer males with darker manes, and they scored this by the number of approaches on the big stuffed lion. So some people think lion manes might be an example of intersexual selection, but other people argue that they might be an example of intrasexual selection, because if you're a lion fighting another lion and he goes to grab your throat and, kill you or what have you, but he instead grabs a big tuft of fluff, that would be a way the mane could benefit them with intrasexual selection. So sometimes it's hard to be sure whether something is intra or intersexual selection. It requires a bit of research. If you want to read more on the lion paper, I posted a link up at the top of the slides there for you. Here's more examples of non-random mating. So assortative mating or disassortative mating are other examples. In assortative mating, organisms with similar genomes and phenotypes get together, and in disassortative mating, different genome and phenotypes will get together. So sexual selection does happen. Sexual selection is a type of natural selection for mating success. It can result in sexual dimorphism, which is when there's marked differences between the sexes and secondary sexual characteristics. So like a male peacock looks a lot different than a peahen, right? They're both in the picture below here. And so that would be a good example of sexually dimorphic organisms. Or even a buck and a doe look pretty different, right? So that would be sexually dimorphic organisms there as well. A lot of times people get confused on this, or they're like, oh yeah, great, I got it, and then they don't actually have it. So be careful on that one. Make sure you remember this, or take the time to remember it. So if we go back to the Hardy-Weinberg conditions, there's math for this because math makes everything better, right? Don't worry, we're not going to do the math problems on our exam, but I will kind of walk you through some things here. So with the Hardy-Weinberg equilibrium, P is the proportion of one allele, let's say big A, and Q is the proportion of a different allele, let's say little a. So P plus Q is 100, and P squared plus 2PQ plus Q squared is 1. I said 100 because it's 100%, so 1 or 100%. This version of the equation only works for diploids. So what's a diploid? An organism with two chromosome sets. So we're diploid organisms, and this only works with a gene with two alleles. So a gene with only two alternate forms. So if it only has two alternate forms, the proportion of big A plus the proportion of little a would equal the entire proportion. So it would equal 100%. And then this next part, P squared plus 2PQ plus Q squared equals 100%, might be a little bit tricky. But you've already done this before. You guys have done Punnett squares before. So if we took big A, little a by big A, little a, you'd get big A, big A. That is your P squared, right? So there's one P squared, one big A, big A. There's two PQ because there's two big A, little a's. And then there's one Q squared because there's only one little a, little a. So if we sum up all the critters that are big A, plus all the critters that are big A, little a, plus all the critters that are little a, we get all the critters in our population. So we get 1 or 100% there. So that brings us to microevolution. We said the Hardy-Weinberg equilibrium said that change in allele frequency wouldn't happen if all those conditions were met. However, we don't meet all of those conditions in a real population. So change in allele frequency over time is all the evolution is, and microevolution is specifically looking at changes in a population's gene pool from generation to generation. So the things that would make that happen are departures from that Hardy-Weinberg or those Hardy-Weinberg conditions. So mutations, do mutations occur? Absolutely. Gene flow, does gene flow occur? Absolutely. Critters enter and leave populations, or gametes enter and leave populations all the time. So mate selection or non-random mating, that absolutely happens. Genetic drift, meaning chance events. Do chance events happen? Yeah. Floods happen. Catastrophic fires happen, right? Mudslides happen. Chance events occur. And then also natural selection, which we'll talk about a bit more coming up here. If you look at the link below, that is kind of a little flash diagram that shows you what happens with all of those things in a population of like colored dots there. So it just illustrates mutations, gene flow, all of that, with colored dots representing allele frequency. It's probably worth taking a look at. So number 4 then, would you expect a typical population to be at Hardy-Weinberg equilibrium? Would you? I would not. So no, you would not, because it doesn't meet those Hardy-Weinberg conditions. Mutations occur, gene flow occurs, mate selection occurs, natural selection occurs, and kind of so on and so forth there. Number 5, give an example of intrasexual selection. So intrasexual selection is within the same sex. So this is like male-male competition. So anytime males are fighting over access for females, that would be an example of intrasexual selection. So I said we'd get back to natural selection, so we're there now. Natural selection is a process in which beneficial traits that are heritable become more common in successive generations. Over time, natural selection results in adaptations, which are just changes in populations of organisms that promote their survival and reproduction in that particular environment. When we talk about natural selection, it's important that we talk about fitness. Fitness in biology means something different than fitness usually means, I would say. In biology, fitness is a measure of reproductive success. It is basically the relative likelihood that a genotype will contribute to the gene pool of the next generation as compared to other genotypes. Normally, when you think of fitness, you are thinking about like how fast you can run or how much weight you can lift or what's your resting heart rate, those kinds of things. And those might change your likelihood of contributing to the gene pool of the next generation, or they might not. So be careful and realize that in biology, fitness is a measure of reproductive success, and it's not so much about how much you can bench press. So for natural selection to occur, a population has to have alleles that vary, and that variation arises from random mutations. Then some alleles encode for proteins that enhance an individual's survival or reproductive capacity compared to other members of that population. And then individuals with these beneficial alleles are more likely to survive and contribute their alleles to the gene pool of the next generation. And then over many generations, the allele frequencies of many different genes may change through natural selection, which would then alter the characteristics of that particular population. So there's patterns of natural selection. The 3 patterns we're going to talk about are directional, stabilizing, and disruptive or diversifying selection. And we're going to go over each of these on the next couple slides here. So with directional selection, this is when one extreme is selected for. So here we've got a population of white mice with a few brown mice. And for whatever reason, some selective pressure favored brown mice over white mice. So it's moving towards one extreme or another. Perhaps it was originally a snowy habitat, and then, there's not as much snow, so now the brown mouse are favored. With stabilizing selection, this is going to favor the survival of individuals with intermediate phenotypes. So in this case, extreme values are selected against. So in the graph here, they've got birds and the number of nests that they found with the number of eggs. And in this case, after many generations, the number of eggs selected for was kind of that intermediate amount. There's some risks with having too many eggs or too few eggs. If you have many offspring, it is a challenge to take care of all of them and make sure they are all fed, right? So that's the con on that side. But the pro might be that, like, even if a snake comes by and eats, 4 of them, if you had a whole lot of offspring, there's still going to be some left, right? But maybe that's more likely that your offspring are not going to make it because you're out, constantly getting food for them instead of supervising them. So having too many offspring comes with some challenges. Having not as many offspring, maybe it's easier to take care of them. There's only one mouth to feed, right? But if that snake comes along and chomps that baby bird up, that was your chance. Right, there's only one, there's no more. So selecting for this intermediate number of eggs seems to give this particular species of bird the best advantage and the best ability to survive and reproduce. So with destructive or diversifying selection, this is when two or more different genotypes that produce different phenotypes are favored. This is likely to occur in populations that occupy a heterogeneous or not evenly mixed environment. So in this particular example, we've got two phenotypes of snails, one that does better in a wooded area and one that does better in a grassy area. So if they are the same species and can interbreed with one another still, but we're selecting for either kind of the rougher shell or the smoother shell, that would be disruptive or diversifying selection. They're selecting for two extremes there. But it's important to be sure that the members are the same species in this case. So let's look at an example here. We've got a graph of the birth weight and infant mortality. This is a little bit dated, right? It's from 1997. And the data is from a study from 1951. So I'm not saying that this is exactly how things are today, right? But generally speaking, if we look at this graph, look at where infant mortality is lowest, right? And look at where birth weight is. So in this graph, it's showing us that most babies are born kind of in that middle range of birth weight, and infant mortality is lowest there. But I think you kind of already knew that, right? If babies are too small when they're born, their mortality rate is going to be higher. If they're too large when they're born, same thing, mortality rate is going to be higher. So this is an example of stabilizing selection. And then number 6, in biology, what does fitness mean? Fitness is a measure of reproductive success. So natural selection can't create perfect organisms, mainly because selection can only act on existing variation. Natural selection doesn't create mutations. It can only work on existing variation. Like, it would be great if I could be tall enough to reach the top shelf in my kitchen. Try as I might, that cannot happen. If I had kids and I had, hopes and dreams of them being able to reach the top shelf in their kitchen, my husband's not very tall either, so they're probably not going to be able to reach the top shelf in their kitchen without a stepladder either. Also, evolution is limited by historical constraints. Adaptations are often compromises, and chance, natural selection, and the environment are going to interact with each other. So we don't create, or the environment doesn't create perfect organisms. It creates kind of good enough organisms, good enough to survive and good enough to reproduce. So keep that in mind. So that was the end of chapter 21. We talked about evolution. So evolution is just the change in allele frequencies. In particular, this chapter we focused on evolution in populations. We talked about heritable variation and mutations. So mutations are just a change in the nucleotide sequence of a gene, and only heritable variation is going to be passed on. A population is just a group of organisms of the same species in a particular area. Some alleles can become fixed due to genetic drift. We talked about the Hardy-Weinberg equilibrium, which we don't ever meet because of the conditions, including natural selection, which actually happens, genetic drift, so chance events, changing allele frequencies actually happens, gene flow, organisms entering and leaving populations, that happens in real populations, and then natural selection and sexual selection, that happens in real populations, so we're never going to meet those Hardy-Weinberg conditions. Why did we spend so much time talking about Hardy-Weinberg conditions if we never meet them? It's a way to show us that evolution is actually happening in a population, because we can compare the gene frequency from one generation to the next, or one generation to 10 generations later. Also, there's no perfect organisms because we can only work with what we already have. Evolution only works on existing variation. It does not create new variations. So that's it for 21.